Hello friends, I am Gaurang Makwana. I welcome you all to my another video tutorial of a biotech guide. And in this video lecture, we are going to discuss about PCR, the DNA copy machine. Imagine needing just one page from a book, but making millions of copies in a minute. That's what polymerase chain reaction does for DNA. So in this video lecture, I'll cover the first introduction history part, then after basic principle with the simplest understanding, then after requirements for performing the reaction, what are the basic requirements are there, then steps which are involved to perform complete one cycle, and at the end application of a polymerase chain reaction. So as usual, today's video going on a very interesting level. Stay tuned with us and enjoy this video lecture. Let us brief the introduction or history part. In 1983, Dr. Carey Mullis developed the PCR. In 1985, first publication of a PCR by Cetus Corporation appears in Science. In 1986, purified Tech polymerase is first used in a PCR, which is a great inventory. Then in 1988, Perkin-Elmer introduced the automated thermal cycler. And in 1809, Science declares the Tech polymerase molecule of the year, which is also a great inventory, and that makes a PCR reaction in a very simplest or easiest way. Let us discuss principle. PCR is an in vitro method for enzymatic synthesis of a DNA. Here in vitro, that means always performed in the lab environment. Then enzymatic synthesis, so here one enzyme is used that catalyze the whole process. That's why defining as a enzymatic synthesis. Now let us understand with the simplest example. So this is our DNA strand, and this is our target part which we want to multiply. So three steps are there: denaturation, annealing and extension. So in denaturation, what happen? This dsDNA, double stranded DNA, converted into the ssDNA via breaking down of a H bond which present between a nitrogenous bases. Then next, annealing part. So in annealing part, primers are there, short oligonucleotide sequences 15 to 20 base pair, which identify the target part or bind them. So here in annealing part, forward primer and a reverse primers are bind. Then in extension, DNA polymerase is also bind at this side and catalyze the elongation process. Here in the reaction, we add free DNTPs in a tube. Free DNTPs, NTPs, that means a nitrogenous bases, which is bind this site. Okay, we are using DNA polymerase or catalyze the sequence. Now number of target copies double in each cycle. So this is our target copy, target DNA is there, which is generated these kind of a copies. Okay, so number of target copies double in each cycle. How we calculate copies of a target DNA by each cycle? So here one formula is there for counting copies of a target DNA is equal to 2 raised to n minus 2n, and also variable length of a target DNA is count by 2n. Here n stands for a number of cycle. So here one example is there. If we perform a 5 cycle, so the copies of a target DNA is what? 22, and the variable length of a target DNA is 10. Assume this is our PCR tube, and in this PCR tube we required a DNA template, primers, DNA polymerase, DNTPs and buffer for performing one single reaction. Now let us brief these all requirements. First, DNA template. So single stranded or double stranded DNA or RNA is needed. If our starting material is RNA, so first step is what? To create a cDNA by using reverse transcriptase. Then if high molecular weight genomic DNA is there, so digesting DNA with a rare cutting restriction enzyme. So these are the DNA template requirement. Then after primers. So primer should be 15 to 13 bases in a length. Base composition should be 50 to 60% of a guanine and a cytosine is required. Then the main and important primer melting temperature is defining as a Tm. Between 55 to 70 degree centigrade are preferred. And this is the formula for calculating the Tm melting temperature is equal to number of A plus T, here A stands for adenine and T stands for thymine, into 2 plus number of G plus C, G stands for guanine and C stands for cytosine, into 4. So this is the formula by calculating the melting temperature. And annealing temperature is equal to Tm minus 5 degree. Tm, that means melting temperature. So first we have to calculate the melting temperature, and in this melting temperature we subtract the 5 degree, and at the end that temperature is preferred for the annealing process. Long runs with a more than 3 or 4 of the same base should be avoided. That means if any basis is continually attached with one run, like if the sequence if base is like AAA, so that should be avoided. Okay. Then palindromic sequence should be avoided. Palindromic sequence, that means the same sequence in a reverse form, like ATGC is there, so CGTA, that is a palindromic sequences, that is also avoided. Then avoid the secondary structure, like hairpin loop. Primer should be not contain sequence that are complementary to each other. So these are the requirement for the primer. Annealing temperature, then long run base pair avoided, palindromic sequences, and a secondary structure. So these are the main factors that are affecting primers or the annealing process. Then third, DNA polymerase. So initially DNA polymerase is a hit label to be added at the start of the each PCR cycle. Tech DNA polymerase, commonly used, isolated from the bacteria, found in a hot spring known as a Thermus aquaticus. That's why the Tech polymerase is name defined Tech, that means a Thermus aquaticus. It works optimally 72 degrees centigrade and over the pH range of 8 to 7.5, adding nucleotides per second to the primer under the reaction. Okay, so here pH is also be maintained 8 to 7.5, and the temperature is optimally works at the 72 degrees centigrade temperature. That is also we set it down in the thermal cycler in the extension step. Then one drawback of a Tech DNA polymerase is what? Lack of a 3' to 5' exonuclease activity is also defined as a proofreading, which can lead to misincorporation of a nucleotide 1 in a 9000 base. Okay, one drawback is there, but is also happening 1 in a 9000 base. If we multiply 9000 bases, so rare chance is there to become a exonuclease activity or a proofreading which cause a misincorporation, but it's a rare. Then deoxyribonucleotide triphosphate, also defined as a free DNTPs. Each DNTP is used at the concentration between a 50 micromolar to 200 micromolar. Higher concentration encourages misincorporation by the DNA polymerase, so optimum concentration maintained. And at the end, miscellaneous factor like pipettes, heating block, thermal cycler, vials, PCR tube, then repeated freezing and thawing block, and inhibitors and enhancer of the PCR. So these are the basic requirement for performing one single cycle. Conventional PCR involves 25 to 50 repetitive cycles, with each cycle comprises 3 sequential reactions, which, as we discussed in a principle, denaturation, annealing, and extension. Denaturation. The reaction mixture is heated to 95 degrees centigrade for a short time period, about a 15 to 30 seconds, to denature the target DNA into a single strand that can act as a template for a DNA synthesis. Okay, so as we discussed, dsDNA is converted to the ssDNA by breaking down of a H bond, and the temperature is 95 degrees centigrade. These are important points. Then primer annealing. The mixture is rapidly cooled to a defined temperature, which allows the two primers to bind to the sequence on each of the two strands flanking the target DNA. Flanking, that means a backside. So the mixture is rapidly cooled down, okay, 95 to 55, and which allows for a primer binding. So two primer forward and reverse are there and binds at the target DNA site. As we discussed, primers are a short single-stranded sequence of a nucleic acid, usually 20 to 30 base pair, selected for specifically hybridized to particular nucleic acid target, essentially functioning like as a probe. Forward and reverse primer sequogrinds the binding site, two strand, melting temperature Tm is equal to formula as we discussed, and annealing temperature should be Tm minus 5, usually 55 around. High annealing temperature reduce the yield and a low annealing temperature cause non-specific amplification. So temperature should be maintained. Then third, extension. In this part, DNA polymerase binds to the free 3' end of the bound oligonucleotide and use DNTPs to synthesize a new strand into 3' direction. Similarly happen in the another side, and performed at a 72 degrees centigrade for a 30 to 60 seconds. So these are the basic important steps for performing a PCR cycle. Now application of a PCR in medical diagnosis detects disease like a COVID-19, HIV or a tuberculosis by finding small amounts of a virus or a bacteria DNA. Forensic science identify people from tiny DNA samples like from hair or blood at a crime scene. Genetic testing, check for a genetic disorder or a mutation in someone's DNA like RFLP, restriction fragment length polymorphism. Okay, this is also a kind of a genetic testing. Then research helps scientists study genes and a DNA sequence in a detail. Then paternity testing confirms biological relationship using DNA. Food safety detects a harmful bacteria or GMOs, genetically modified organism in a food. Environmental monitoring finds specific microbes or pollutants in a soil, water or air samples. So these are the application of a PCR. So here we completed our video. I hope you enjoying very well and got it very well. Thanks for watching, and if you want to get more and more updates regarding like this, join with us and be a perfect biotechnologist.